So I have to be super clear that what I'm trying to do is extend the sort of healthy portion of lifespan. Humanity is like intrinsically wonderful. Make it, for me, feel a little bit more like really team human, which is a really good feeling. It's a positive emotion for me. I think once there are really powerful, large effect sizes, therapeutics that can work on aging more generally, the regulators will figure out how to get out of the way. I think much more impressive stuff gets built when people's discount rate drops, and they're not just living for tomorrow.
Body. What is it?
Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as penicillin.
The question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
How would you describe like your philosophy about life and lifespan extension? Like, why is this something that more people should care about? And I am obviously someone that cares about it, but there's a lot of people that see it as sort of this billionaire's side quest, right?
Yeah, well, that's like three different things wrapped up into one. Philosophy on life. I can try to encapsulate that into as short a response as possible. Maybe like it's super fun, and it can be extremely rewarding if you do it right. And it seems to like get better the more you do it, as far as I've noticed. Obviously, there are a lot of experiences that people are having in their lives that aren't so great. Often, when I talk to people about how I want to extend lifespan, that they're like, why? Like, it gets to be such a horrible place for people. Like, why would you want to drag that out? So I have to be super clear that what I'm trying to do is extend the sort of healthy portion of lifespan. I guess healthy there is code word for meaning that you're actually having a good time. So I think that's why I like to do it. I think people are really cool. It's like part of what makes the whole journey exciting and beautiful.
I agree. I watched some of your one of your TED Talks that you gave on your website, and I agree with the philosophical argument that aging and lifespan extension are probably among the most important problems that anyone could work on. They cause an immense amount of suffering for humankind. Age-related diseases are a huge burden on the healthcare system. There's just a lot of reasons to advocate for doing this. And the main challenge is, like, how do you actually do it, right? Because it's been a hit or miss industry. I think like longevity as a word has been associated with sort of like the longevity bros in SF and sort of taking a bunch of peptides and, having a Chinese peptide dealer and et cetera, et cetera. But, there's a difference between that form of longevity and lifespan extension scientific research, which is what Retro Biosciences is doing. And I think a good place to start would be, how did you decide that the mission of the company should be to add 10 years of healthy lifespan to humanity?
Oh, specifically 10 years.
10 years.
There's pretty much no other answer that's reasonable. Because if I choose less than 10 years, it's just kind of meh. It's not that inspiring for people to join in. I can't obviously do it alone. It's like massive team sport that we're engaged in. Like, biology is super, super hard, and lots of different specialties have to work together. But beyond that, it sort of gets a little bit more into fantasy land. If I say, like, yeah, I'm adding 30 years to human healthy lifespan, people are like, how? Do you have any clue? It's like, well, no, there's faith. I guess there's probably a bunch of stuff that'll work. And I think there's a whole I mean, if you put it on one scale, that maybe all of sort of biotech and specifically that which relates to extending healthy human lifespan, there is a spectrum that you can put this on, where there are the hyper conservatives at one end who are like, if there's one sort of accomplishment of humanity, it will be like, I don't know, curing the common cold or something incredible like that, which obviously would be a great thing. But and then at the other end, there are the people who like fervently believe that they're going to create immortality. And it's there's sort of like, both ends are kind of like not so interesting to me, because one's like sort of abandon all hope for the incredible things that technology will bring humanity, which are many in my opinion. And the other side is sort of abandon all data. And in the middle is the area that's most exciting. Like, pick hard challenges that you can actually accomplish. That's amazing. So 10 years, we can definitely do that.
Okay. In what time frame?
Well, if I were perfect at predicting the future, I'd probably be in the wrong business, and I should be probably in finance or something. We've done north of 3,000 experiments so far in the company. Just crossed it a while ago, based on our sort of electronic lab notebook system that records them all. The reason we do experiments is because we don't know what the answer is going to be. So you can pick general areas in biology and knock them off, kind of like engineering projects. Like a lot of the things that happen especially around Silicon Valley, I bucket into engineering projects where you decide, we're gonna build a website that's be able to serve apps to a phone, and then you're going to be able to swipe left and swipe right and be able to choose, whether you like or dislike something. And like, it may not be built yet, but you can build it. That's engineering. Like, we're going to make a rocket that has twice the payload of the last rocket we built. Okay, great. People have been building rockets since the 50s. We know how to do that. It's an engineering project. Biology is a little harder to find things that are pure engineering projects, because it's just so much we don't understand about it yet still. But you can still create little bundles that, like, you're pretty sure you can do it, like high probability. But even still, when people get even feeling pretty confident, they're going to do a clinical trial. You have to try it out in humans. You can't just start selling it. And that's because we actually don't know until we do the trial. But we have a long history of having pretty high confidence in something, trying it in, say, mice, and seeing like a reasonable probability of its ability to translate from proof of concept in mouse to functioning in human. And after a while, you can get a feel for it. So we've extended the lifespan of mice a bunch of times now, specifically on the academic side, such that, you could see how if you add all those together and also sort of divide by the ratio at which things that work in mice don't work in human, you can get to 10 years. I mean, it's a bunch of work. But if I wanted to exactly predict it, then you should just ignore the rest of everything I'm going to say, because there's no way I'm going to predict the future to that accuracy. But I don't know, 10 or 20 years.
Okay, understood. I guess if you had to take us back to the founding of the company, science as an area of, as you mentioned, is not an engineerable problem, right? You have to pick your areas of scientific inquiry. You have to plan the right experiments and create the right hypotheses. How did you decide on the three core areas that your company is prioritizing and working on, when the lifespan extension field is so vast, right? There's people working on peptides and small molecules for genome integrity. There's people working on gene therapies. There's people working on reprogramming, which is something that your company is also working on. But how did you pick these particular areas of inquiry?
So we have taste and we have criteria, and we also have mistakes, the ability to, like, the huge actual luxury, financially speaking, to be able to make mistakes. So these aren't the first three that we started with. We've tried a bunch, more like 6. These are the 3 that checked all the boxes that with a high enough probability to take to the clinic. But even at the outset, we have criteria to apply. Like, is there some robust proof of concept of the principle already, especially in mammals? If it were to truly work, would it be ultimately usable preventatively? Does it actually address an aging mechanism? There's a whole list of things we apply that ends up making the possibilities out there narrower and narrower. We also really want strong effect sizes. So if there's something where you'd have to maybe take some therapeutic all your life to get a moderate effect at the end, then it's a little bit too weak of a sauce for us, because it's largely not because we don't think it would be valuable for humankind, but mostly for practical reasons. It's very difficult to execute a clinical trial and get a significant answer within venture timeframes. Or if you do a clinical trial for 6 months, a year, 2 years, stretching it, 3 years, starts getting painful. So it has to have a reasonably large effect size. That's one of the other criteria.
Mm-hmm. So the 3 broad kind of areas that your company now focuses on is blood plasma, reprogramming, and autophagy. But you mentioned there were sort of 6 to start with. Like, how did this sort of whittle down to what you're focusing on now?
Yeah, so blood plasma, we decided wasn't going to end up being a strong enough effect size for us to work with.
Okay.
So we are still working on blood, but we're working on replacing blood stem cells in the body with replacing old ones with young ones, which then doesn't necessarily directly affect the blood plasma, but certainly creates a new source for all the rest of the blood, which is a lot. It's 80% of the cells in the human body, of the human cells in the human body, come from blood stem cells that are in our bone marrow.
I didn't actually know that. Okay.
Yeah, it's kind of freaky how much they percolate through every cubic millimeter of our bodies constantly.
Mm-hmm.
It's cool, but yeah.
Yeah. So I guess I'm curious about the sort of areas that you started off with that you sort of failed to validate, right? Sort of whittling down. Can you share a little bit about that?
Yeah, sure. So for the blood plasma thing, there was interesting proof of concept in mice, and we explored it pretty far, and to the point where, in more complex models than mice, I guess I'll say, the farther we went, the smaller the actual effect sizes turned out to be, such that it was converging on, compared to how kind of heavy duty the intervention is, basically having to run your blood through a circuit outside the body and back in, through like some very large, scary needles. With the concomitant risks of infection and various other breakdowns for these complex machines, didn't seem like it was going to be deployable on a large scale and frequently, but maybe it could be justified if the effect size was really large, which it didn't turn out to be.
Okay, understood. Now, I think one of the ways that your company is obviously very well known for is having raised quite a large amount, about $180 million from Sam Altman. That's the big splashy headline that drew everyone's attention to retro in the first place a few years ago. And I guess everyone has wondered, like, how did this company attract so much interest from Sam? Like, how did you build that relationship? Obviously, you were partners at sort of YC, or he was a partner at YC. I think you worked at YC. How did that relationship form, and what gave him the confidence to take such a big, bold bet on you as a scientific founder?
Yeah, well, I was a part-time partner at YC. Sam was president. I'm not going to speculate what was going on in his head, but you can ask him if you want. I think it was pretty clear we both had an interest in these kinds of areas of biology. Just empirically looking around at the things that Sam's taken big swings at, they are things that are technically hard and that have potentially huge benefits for humanity. So, fusion power, obviously, energy is a giant pain in the butt for humans right now. All kinds of regional disagreements and climate, depending on who you ask, disturbance on the climate, and that's worth going after. AI obviously can have a big effect on huge amounts of human drudgery that goes into things that are kind of boring. Having to do boring things is a kind of violence almost. And dying of age-related diseases is also like pretty heavy metal. So, okay. I mean, there are a few things like that. I would love it if people would stop having wars, because I think that's a big source of misery too. I have no idea how to do that. So I like to focus on really difficult technical things that I have an idea how I might approach them. That one's like,
Take us back to the early days of the company, right? How did you arrive at this hypothesis on extending the healthy human lifespan? Why did you feel that it was sort of your moral imperative to do so, and B, something that you were going to be particularly good at, or sort of have a high alpha in your ability to get it done?
Yeah. Certainly it happened before Retro. It's like probably right near the end of my first venture funded company, I ran into a book by one of the actual legit academic aging biology researchers, Roy Walford. And because I was interested in health generally, of course, and I don't think I'd call myself a longevity bro at that point, because I'm not sure that stereotype existed, but the body is super fun, so of course I want to keep the body in good shape. And but it didn't seem like there was really, like, if you look around, certainly then if I looked around and said, okay, what's the advice? Like, how do I become a responsible steward of this biological blob that I call home? Half the people are saying, eat just raw red meat or whatever. The other half are saying, no, don't, eat any red meat. You want to, like restrict methionine and have a low protein diet. And, there's so many different diets, and there doesn't seem to be anybody at the top level of that hierarchy saying, okay, these guys are right and these guys are wrong. There's just disagreement. And it seemed kind of intractable. Like there's no solution to this. Just give up. But one thing I liked about reading Walford was that he made it really clear how you can collect definitive data. I have a background as a scientist. Of course, I want to be able to conduct an experiment and say, yeah, this thing works, this thing doesn't. That can guide action. And he made it really clear that you can do research on humans and you can test whether things work or not. And there's this very clear, beautifully structured, statistically valid ways of conducting experiments. And there's retrospective, cross-sectional, prospective, stratified, randomized, double-blind. There's this sort of an echelon of like better and better human evidence where you can ultimately actually make conclusions. And I thought, this is a realm where you could actually bring science to it, and it's not just a bunch of people trying to yell commercially louder than each other to sell you their things that probably don't work. So that kind of got my imagination going. Like, this could be a realm.
Mm-hmm. Okay. But how did this turn from like an interest in sort of dispelling the disagreement around like nutrition to this like focus on?
After that, after I guess a number of things came together. After the liquidity event for my first company, then I was sitting on the beach, kind of like, yeah, done. This is good. I'm just going to chill here forever. And then it started getting kind of just feeling sort of sad, just feeling kind of empty and boring and like, okay, I have to do something. And what do I want to do? That lasted about 10 days. It should be something that at the end of it, I don't feel similarly about. Like, what did I actually accomplish here? Not that much. Made a bunch of cool gadgets that are going to be heading to the landfill when the next cool gadget comes along. And so that was happening in me at the same time as Roy Walford echoing through my head. And I think the idea of just sort of sitting there on the beach, getting older and older, and it's like doing jack, just also kind of grated on me. And then, so I guess also after reading Walford, like listening around in the valley to people talking about aging biology, or not so specifically aging biology, but longevity, it was terrible. Like the dialogue was all about like buy my vitamin thing, do my expensive vitamin IV, or take whatever, put this cream that supposedly reprograms your skin or whatever, and it was all terrible. Like if you read any of the evidence behind it, it's like vapid, and that, I guess part of it is like we should be able to do better. And so I created a nonprofit that started bringing in academics from the sort of university version of the study of aging biology, which, was like Walford's lineage of people who use real data and evidence. And I just decided, right now I'm going to work on leveling up the conversation in Silicon Valley and see what happens, which was fun. I met probably 24 pretty badass researchers, professors from universities all over who study aging biology.
This was pre-Buck Institute or sort of?
No, the Buck was already there.
Okay.
This is starting in about 2012. A few people from the Buck came and spoke at some of these events. I held these big public events where lots of people would come, and a few companies were formed out of them too, and at least one clinical trial got spawned at one. I was like, oh, I feel proud of that. Judy Campisi came down and talked. May she rest in peace. Cynthia Kenyon, amazing. She's a legend. She kind of kicked off the whole academic field in a way. There's a much older one of people who figured out that calorie restriction will extend the lifespans of rodents, et cetera. But it really got going around early 90s, like 93, when Cynthia in her lab figured out that you can tweak one gene in an experimental animal and double its lifespan. Just one single change like that, which got a bunch of people who are thinking, what should I do my PhD on? Or where do I want to do my postdoc? Thinking, oh, wow, it's that mutable? Because before people kind of thought like, well, it's just this sort of inevitable entropic wear and tear process. You can't buck the second law of thermodynamics. Then it's like, oh, wait, it's actually malleable. If you can tweak one gene and double it, there's probably a bunch of other stuff you can do. And since then, I think it's now up to like 10x lifespan for those particular kind of worms. Yeah, she came and talked, and it was good. It just got like very steeped in the actual world of how that stuff is studied.
I think even just within my shorter lifetime than yours, the conversation and the enthusiasm around lifespan extension longevity has probably around 10x'd. Like when I was growing up, there were like some inklings of people talking about, creating lifespan extension companies and healthspan extension companies, but no one had taken a drug through clinical trials. People weren't really, the conventional CRISPR scientists weren't talking about aging, right? Mm-hmm. Now, if you look at sort of the Arc Institute and you look at all the the George Churches and the Omar Abadayas and the Jonathan Gutenbergs of the world. Like, this is the thing that everyone wants to do. There seems to have almost been like a tipping point that this is actually like a tractable problem, as you said, and that sort of if we actually allocate collective societal resources towards the problem, that like we'll figure it out, or at least we'll make a substantial dent. And I think the thing that I'm curious about is sort of in what directionally, what kind of a dent do you think you will make? So you have these 3 programs. I want to understand sort of what the progression is of these programs, how they're going, what you've learned, right, over the course of the company's existence on the therapeutic side.
So on the therapeutic side.
Yes. Okay. I narrowed it down towards the end. Yes.
Thank you. Yeah. So as I've mentioned, I have a bit of a bias toward things with large effect sizes. So I'm not saying, hey, let's take metformin and try to get it through a trial, because I think it's only going to have a pretty small effect on healthy human lifespan. You know, something, but just like a weak sauce. So what we're working on right now are things that can make a difference for huge diseases of aging. They're not specifically focused on these diseases of aging from a mechanistic perspective, but they would and should theoretically have a big therapeutic effect on them. Specifically, Alzheimer's for two of the therapies, and age-related loss of immune function for the third one. So Alzheimer's it's going to be like a trillion a year, something, just in the U.S. If we don't do something about it. There's nothing that really works very well right now. There's like one drug that like kind of works a little bit. So our first thing into the clinic, which should be in like first dose a little bit later this year. It's starting to be weeks now instead of months, which is great.
This is the clinical trial that you're running in Australia?
Yeah.
Later the year? Yeah.
Yeah. So that if mice are anything like humans, which they are somewhat like humans, just not unfortunately identical. Actually, it's probably fortunate that they're not identical. Then we'll be having a big effect. And that works by also something that is a word that's massively overused by, as you call them, the longevity bros. I like that term is really fun. Like autophagy, which is, it's the thing that happens when you fast, right? Well, it turns out it's a extremely complicated sort of whole realm of cellular biology, and there's a number of different autophagy pathways, and different stages of autophagy pathways, and et cetera. But there's certainly one of them that gets messed up in the later stages of especially neurodegenerative diseases, and that's what we've found, a classic small molecule drug that reboots that. So that's one of the 3.
Hey, by sort of talking to retro engineers, I mean, I lived in San Francisco for a year. I was chatting to some of your sort of team members at the Bits in Bio events a while ago, and one of the things that they said about you was that you're very, very obsessed with just getting these drugs through the clinical trials. Like it's this like good hyper fixation and focus on like the actual end state where value is generated and sort of where impact on human health is measured. And this is something that, in their experience, some other sort of biotech founders struggle to do. They sort of become too obsessed with the big picture, the sort of grandiose ambition, and they kind of delude themselves with the grandiosity of the ambition in some kind of like a intelligent way of self-sabotaging, right? And for you, like, in a relatively short time span, albeit relatively well-funded company, you have brought like genuine drugs into clinical trials, or at least sort of commencing
very soon.
Very soon. What is it about the way that you structured the company, about what is it about the company and the way that you thought about like founding it and building it that allowed this to actually happen? Like when people say you're really focused on getting a drug to the market and just getting it to clinical trials, getting something to clinical trials, how does that manifest in your decision making? Like, what are the actual decisions that you would make?
I mean, it's going to be a little bit hard for me to introspect on because, of course, it's just, well, this is the way you do things. So it feels implicit to me. But if I try to step outside and look objectively and dissociate a little bit, I guess. For one, I think I have a pretty strong kind of bottom-up philosophy about how to do things. So I like to build. There's some sort of, like, I guess in some ways, like impatience. I like to build a thing and have it work, and that has how I've succeeded at many of the things that I have attempted in my sort of so far weirdly meandering and long career. Just go out and build it. Get a prototype of it working. Okay, it's possible. Okay, now make it, harden it, get it so you can actually ship it. And so I like to push through at least one vector all the way to some kind of endpoint, just to say, yeah, we know how to do this. Because I could work on 5 things at once in parallel and slow all of them down and not have any idea really how long it's going to take. So I think biology is really, really complicated. Aging is also extremely complicated. And so I think it's going to take a bunch of parallel efforts to achieve our mission. So I don't want to just make retro into this sort of binary bet on one specific mechanism, because we're going after a mission, not a specific disease or platform. But I want it to be the smallest number that we can make rapid progress on. So I think also, to me, time is one of those really like core variables. That it's a variable that it's just one of the things, you set the temperature in the room, it has to be right, or people can't function in there. It's like one of the key, how do I put it, like solving for how we move fast is one of the core, like, sort of venture engineering parameters, because everything else relies on it. If you go slow, you run out of money because you're burning most of your money on salaries. If you go slow, it's boring. People aren't as enthusiastic. Not as many people want to join, because we want to have a place where we're making so much progress that it's exciting to be there, because of course we want to have the absolute most capable people in the entire field wanting to join in with us and work together with us on this mission. Mm-hmm. So it has time is one of these variables that has all these compounding benefits to doing well at it.
How do you tune the temperature?
One of the things that slows people down is that they try to make sure they're not going to screw up, because they probably got a fair bit of like negative feedback for screwing up in the past. So they'll try to be too careful, and they'll design before they build, and so that when they finally build, it turns out perfect.
Sorry, when you say build, do you mean test?
Everything is building to us. We have this giant sign on the wall of the sort of the original lobby, the company, that says T2B on it, which means it's time to build, which has the word time and building right in it. So building just means executing something that you envision such that it comes to fruition. Getting a particular experiment, like I think experiments have typically not just 2 outcomes, but more like 3. It's like you have a hypothesis, and then you end up finding out, like, is the hypothesis true or is the hypothesis false? But there's this huge middle zone where it's like, we couldn't tell. That to me is failing at an experiment. And whether you get a yes or no answer, that's the data you wanted. But I think a lot of the time people don't really know what the hard things are. I guess that's the hard thing about hard things is you think you understand the problem and you can spend a long time trying to do it perfectly, and then you actually start doing it, and then you realize what the actual hard parts are, and then you have to kind of, in some ways, start over anyway. So one of the things that I encourage people to do a lot is do a reasonable amount of preparation. As long as this isn't like you're trying out a new therapy on a human, as long as it's a somewhat reversible thing. It's like, okay, you just spent 5% of the budget for the year on this first attempt, and it's like, maybe it works really well, or maybe it crashes and burns, and you're like, oh, that's what's hard.
I see.
And then you do it again, and then you do it again, and usually by about the 3rd iteration, things are going pretty well. People have kind of figured out, and so that's just an iteration to us.
Interesting. To relay this back, right? You're saying that people are not risky enough when doing these ventures, that they spend too much time in the sort of experimental design and maybe even the design of like the molecules, et cetera, phase, and not enough on like data collection and just trying things and seeing if it works, and sort of seeing if the endpoint of the assay is sort of positive or negative, et cetera, and that you sort of, by just testing and by moving more quickly, you can identify the difficult parts, right? Just science is just a convoluted thing, right?
An assay is a great example. Like, I think smart people tend to abstract really well, and so they'll want to abstract down to the skeletal, logical bare bones of an experiment, and say we're going to touch these molecules, and then these 2 will bind, and then it'll like inhibit this 3rd thing, and then we'll find out whether it works or not, and then. The thing that the mind is not terribly attracted to is, okay, now we're going to try even the first thing, or we're going to bring up an assay to actually measure the thing that we were expecting, and then the assay just doesn't work. Like, and you try your positive and negative controls, and they just all come out 0 or something. You're just like, why? And then you have to thrash around for a while and realize that, oh, okay, you're supposed to do it at 25 Celsius, but you did it 19 in the lab because it was a little cool, and or whatever, or you end up trying it 5 times and eventually you get it. It's the really boring, grungy things that often slow you down that you don't even realize are going to be the hard part. Sometimes everything just works, but very often you'll find that some of those things, like you're expecting a fluorescent readout to tell you when the protein starts being expressed, but it's like always fluorescent because there's some sort of background, or it never reads out, or then you have to like trash that protocol and try bring up a different assay that gives you the same result. And the sooner you find that stuff out, the better.
Yep, noted. I'm planning my own experiments right now in the lab, and I feel you. I want to switch to reprogramming from the perspective of science and sort of how it's evolved as a field. You had the discovery of the Yamanaka factors, and then you had the discovery of partial reprogramming, where if you sort of pulse them, you could get the cells to return to some kind of a more naive state. What has the progress been since then? Like, what is the arc of history on reprogramming?
Well, it started off as germline modification of animals, so that they're. I guess it depends on what you mean by reprogramming per se, but I think you're talking about partial reprogramming.
That's right.
Okay. Yeah. So like germline modified animals, which means every cell in their body has like some kind of special switch you can turn on and start a reprogramming process, usually with some sort of small molecule outside drug that'll activate a gene cassette. And so I think that's where it started, like from Alex Ocampo's paper in 2016, which is pretty much the first in vivo partial reprogramming that was ever shown. And then it went to expressing these genes only in specific organs to try to understand these life extending or rejuvenating effects. Are they mostly coming from one particular organ or another? If you do too much reprogramming, why does it get toxic? Looks like the liver reprograms faster than most organs, and that's one of the major sources of toxicities in the liver. For instance, so it's been more like, okay, this is an amazing proof of concept, and we've seen a bunch of aging phenotypes reversed or improved in mammals. Now let's get into the nitty gritty. How does it work in different tissues? So like going in tissue by tissue, I think is kind of where it is right now. Like doing it globally is just probably a little dangerous. So we have to start getting really specific about which tissue we're reprogramming in which way and how much, and not just the whole tissue perhaps, but only maybe certain cell types within the tissue, of which there are many in most tissues.
Mm-hmm. And for our audience's sake, how does targeting work, right? When you have organ-specific, tissue-specific, and cell type-specific targeting of these reprogramming factors, what's the actual method of delivery that's specific?
Yeah, I guess there's kind of three ways of targeting. One is very crude, but you can target by choosing where you inject something that causes those transcription factors to be expressed. For instance, if you inject it in the eye, which I know sounds grisly, but there are these tiny little needles, and lots of eye injections happen these days very safely. Then that is targeting the inside of the eye, and for the most part, it doesn't end up going all over the place into the rest of the body. The second would be to use vectors that only go into certain types of cells. So, for instance, a lot of gene therapy these days is done with especially modified viruses that don't just spread throughout your body and cause like the flu or something. They can't really replicate in the body, but they're still really good at that other virus trick, which is getting into a cell. And so it's a way of carrying a medicine into a cell, put it into a virus. And they have fairly specific refined machinery for detecting the cell they want to go into. And that can be tweaked. So you can have a virus that only attaches to cells that are expressing a particular protein on the surface of the cell. And then, inside a body, different ones of ours are different types of the cells in the different tissues that we have sometimes have differently expressed proteins on the outside that can be used as the signal for the virus to grab onto that cell and get sucked in. Mm-hmm. And AAV is a good example of that. There are a lot of very specific cell types or tissue types that different variants of the adeno-associated virus will go into. And then the third, and there's probably more, but these are the three that occurred to me, is once it's in the cell, does it do anything there? And that one way to do that is by the design of the so-called promoter, which is some beginning beginning sequence on a gene where that sort of determines whether or not it's going to get transcribed or not.
So you can put like a cell type specific promoter, for example, to ensure that it only expresses in like neurons, right?
For sure, yeah.
Okay.
So there are different gene programs are turned on in different cells because they're doing different things, and that means different proteins are being generated, and that means certain the machinery that turns on one particular promoter may be active in one cell versus another one.
Welcome to the Frameshifts podcast, where we spotlight the world's most ambitious biotech founders, investors, researchers, and changemakers. This episode is brought to you from the offices of Polsinelli, a national law firm that helps biotech companies do everything from incorporation to patents, trademarks, and venture financings. Thanks to Polsinelli for being part of the growing ecosystem of scientists, investors, and builders shaping the century of biology.
So I'm really curious about how you're thinking about your first reprogramming clinical program, right? I mean, what's the indication? What's the method, and sort of what's the rationale?
The first one we explored was in liver, and somewhat naively also. It was fairly early on for us, but the idea was, well, liver is a very important organ, and there's plenty of liver disease out there, and the liver is very easy to target with the gene therapy because it's kind of like the proverbial side of the barn that it's almost impossible not to shoot. In fact, most failed targeting ends up going to the liver anyway. It's the everywhere else sort of location. But nonetheless, we picked a particular AAV that was good at going to liver, and designed the right promoters to express specifically where we wanted it, and we're actually shockingly successful at partially reprogramming liver in mice. We didn't try it in people, and then partway through that process, the GLP-1 agonist drugs caught fire commercially, and the entire sort of liver disease landscape started rapidly changing because most of liver disease comes from obesity. And so then, we had the decision, should we take a fairly complex therapeutic and take it to market to alleviate certain kinds of liver disease that were similar to the kinds that GLP-1 agonists were modifying, steatosis and so on. At the same time, it's massively changing. And the decision we came up with was no. It's going to take easily 5 to 7 years for it to get to market. Like, what's the market going to be like in 5 to 7 years? Very hard to shoot a moving target like that. And the other kinds of clinical trials to do in liver are actually really hard. For things that are off axis from the types of things that the diet-induced liver disease causes. There's alcoholic liver disease, which is extremely hard to do clinical trials in. There's drug-induced liver disease, virally induced, and none of them lend themselves to trying a gene therapy intervention. But it was a very inspiring proof of concept for us. Like the results were dramatic. Like, we rejuvenated mouse liver for sure, based on multiple readouts, at least 3 or 4 different phenotypes that are age related, got reversed. So that spurred us on internally as, okay, we sort of showed our mettle there in the partial reprogramming and some more therapeutically applicable partial reprogramming. So we're exploring a few different tissues now. with stricter criteria on if we succeed, will there be a huge humanitarian benefit and a practical clinical trial to execute.
Mm-hmm. Okay, so nothing that you can share as of yet.
At the moment, no.
Okay, got it. I suppose the natural next topic would be GPT-4b. So when this came out, I just assumed it was an LLM. It was within the same sort of nomenclature. I thought that I'd be able to use it, but it's sort of specifically for the sort of design of new and improved Yamanaka factors.
No,
no,
definitely not.
Okay.
Well, okay, you said a few things there. One, it was an LLM. Yeah. Two, you could use it. Not so much. Which is not to say anything about your particular skill level. It's just that it's not generally available.
Mm-hmm.
Three, it's definitely not specifically focused on transcription factors or Yamanaka factors as classes of proteins. It's very actually protein general to protein generation. And we just chose Yamanaka factors because they're near and dear to our hearts, and we've studied them a lot over the years, and have lots of assays that we know work. Back to assays are really hard, and often don't work even though they should. So, yeah, why don't we mess with some of those?
So, like protein design generally is the interesting.
And one good reason not to make that generally available is that there are lots of kind of scary things you can do with protein. If you have like a sort of a general protein generative model, you can make new viruses and theoretically poisons and things like that you wouldn't want people to just necessarily be able to do without a lot of like strong security guardrails around it.
Mm-hmm. So explain to me then, like, what is the interface? What do you input? What do you get? Like, how do you specify the design of these proteins? And
so it's yes, LLM, but they're large language models. The language basically means it is trained on a sort of sequential data type, because text is sequential. We read from left to right or from right to left, depending on what country you're in. And that's a particular direction. And turns out proteins are the same. There's a sequence of amino acids, and it matters what order they're in. And they're long, thousands or tens of thousands of letters long. So it's very, very text-like in a lot of ways. We trained a new large language model that had both a text understanding capability and a native protein sequence understanding capability, both joined together. And so that, I guess you asked, like, how would you interact with it? Mm-hmm. So you can, it understands text, you can put text in, but it also understands sequence. And that is a particularly powerful aspect of how to prompt it. So you can put in relevant sequences or sequences that progress in a particular direction as a way of indicating what the additionally generated proteins should be.
Interesting. Okay. So a list of sequences that are sort of directional in some way, where there's modifications made, for example, and it sort of predicts the next modification. Is that accurate?
Yeah. So it'll predict. Sometimes if you're very unlucky, ChatGPT will, instead of giving you its top prediction for the completion of your prompt. It'll give you a couple side by side, and you're like, I have to read both of these and decide which one I like better, which is very hard because they're usually both quite good. But that means under the hood, it's generating a most probable response, and it can generate a bunch of other responses as well. So similarly, this model will generate a list of a bunch of different possible proteins that it thinks are probable solutions to the query. Let's say 100 of them, and then we can evaluate them using a bunch of different criteria we have for how proteinish does it look.
Okay, interesting. Does it have the capacity to also do sort of natural language prompting of, this is a sequence I want to add an additional domain? Does it do that, or are we not there yet?
It's not quite as English.
Okay.
As that. I mean, it can understand English, but in terms of the interactions between the two are mostly come from scientific papers, which are usually mostly talking about other things, or protein annotation databases, which have some text associated with different proteins.
Mm-hmm. Okay, got it. What are you using it for internally?
So far, we've used it to make new programming factors, which actually ended up in one of our recent FDA submissions. So we're feeling pretty confident about its ability to be better at reprogramming. We also redesigned like a cell surface receptor for a specific purpose, which it seems to have done a nice job at. It succeeded in designing in the properties that we were wanting for the receptor. There are some other targets that we're going after now in the aging space that we're trying to design better versions of the human proteins. They're kind of more like ones that perform well for humans that live longer. Different humans have different variants of various genes, and some of them seem to help them do better.
Interesting.
So we're trying to understand, like, why are they better, and how can we make them like way better?
Mm-hmm. Well, I think this is an area of interest that we share, sort of on creating supernatural versions of existing endogenous human proteins to extend lifespan. Any particular areas of interest? Like one thing that I'm super interested in is sort of the DNA repair factors, the high fidelity DNA polymerases, and kind of long-term reinserting these back into the human genome at those natural sites and like genuinely upgrading the molecular machinery that maintains the genome. What do you envisage as some of these more like long-term applications?
DNA repair enzymes are super important. Like they're very interesting from a longevity perspective because they're one of the things that sets apart, let's say, species that live multiple hundreds of years from pathetic ones like human that only live like 0.8 years.
We're pretty good. We're not that shabby.
We're pretty good for our body size. I'll say that.
Mm-hmm.
Like you can make a chart of animal body size and lifespan, and there's a kind of a cloud, but there's like definitely a correlation. Like bigger animals live longer. But then on that chart, at the weight where humans are, they're like way above. We've already, like from an evolutionary perspective, we've already put a bunch of work into trying to figure out how to get this thing to live longer. I assume because it's like very awkward to like we kind of live or die based on knowledge that we have, and it's very awkward to get the knowledge in. Like you have to like hang around for 20 years and like listen to very slow data streams, and eventually it soaks in. So we need a little bit of extra lifespan to get all that training to happen. So that's why I assume that humans have worked at it a little bit harder.
I think you're probably right. We're particularly social species. It makes sense that we operate on longer time horizons, and sort of we have a particularly extended adolescence as well, which is kind of akin to what you're talking about around how long it takes us to learn basic skills. Yeah. So DNA repair interests you as well. What do you think will be like the first therapies that result from sort of enhanced, better human proteins?
I don't think I know yet. It very possibly could be like a retro design protein. Yeah, I think it's possible that somebody might be able to borrow a protein sequence that exists from another species and swap it into human. That might work.
Mm-hmm.
I think there could be some issues around how to, deal with immune rejection and that kind of thing. But it's a pretty tried and true method. Like the way that most of the really amazing leaps that have happened in biology over the last a few decades have generally been from someone going out and borrowing a protein that existed somewhere else and just moving it. Because so far, designing them from the ground up or making major changes on our own is like,
it's tricky.
So intimidating that it's like, ugh, don't even try. So we got PCR working, obviously, because some lunatic decided to see if he could get an extremophile polymerase to work, which amazingly it did. But that was borrowed from a bacterium, and then recently, in the way I see it, we got CRISPR to work, and that's again borrowing an existing protein that existed for another application, also from a bacterium. In this case, so maybe what'll happen is somebody will borrow some DNA repair machinery from bowhead whales or something, and figure out how to get that working in humans. That'd be amazing. But it's specifically kind of that history of like, there's this sort of like learned helplessness, you could call it. I think it's totally appropriate and understandable in the sort of frontier biological realm, where it's like, well, of course we're not just going to like redesign our own protein from the ground up. Like, we're going to go out and use something that's already working out there.
Which is strange, because we do quite a good job of protein engineering natural proteins. Like, usually their efficacy or efficiency is like improvable by orders of magnitude, right, compared to what nature has arrived at through directed evolution. And
yeah, I think in areas where we've been successful reductionists, and like a huge amount of human successes have come from us being like hyper reductionistic, trying to figuring out how to split atoms, and then going first figure it out and then scale it up. So I think that if you really drill in, and people can spend, people spend a lot of time in biology really deeply drilling in and figuring out exactly how a certain thing works. And if you really drill into a particular protein, how it gets its job done, and like there's a pocket where it binds to some metabolite or to some other protein very specifically, and you happen to know that most of its effect depends on this little stretch of 3 or 5 amino acids, or sometimes just 1 or 2, then okay, the combinatorial sort of total possibility sphere isn't that big. It's like 20 amino acids per location for a fully orthogonal test, and if you're doing three of them, 3 to 5 of them, is it's some number of millions of possibilities, but that's testable. So I think you can get things done if you have like a very, very close mechanistic understanding.
The key there is mechanistic. We just lack so much mechanistic understanding of a lot of endogenous proteins.
Yeah, at least in 2025.
Yes. And presumably also creating small molecules and peptides that like bind in the right ways to modulate the efficiency and just activity and fidelity of existing human machinery, I think is another. All of these, I think, are unexplored, relatively underexplored approaches that I think more people should work on. And obviously, I would love Retro. I'm sure you're already thinking about some of these ideas.
I mean, the cool thing about GPT-4b is that it massively changed the proteins in ways that we could just never figure out. You know, instead of tweaking three to five amino acids, proteins are typically thousands of amino acids long. It can change up to 80% of them, and somehow the thing folds as a protein. But then not only that, it like functions, and then a lot of them function much better than the natural ones. So largely we haven't a clue. It's a very non-reductionistic space for us. It's just like, okay, there's deep patterns in there in biology. It's just like we're not really that sure how an LLM understands human language. It's like, where's the rules about adjectives and adverbs and all that stuff encoded? Like, not really sure, but it somehow figures it out. There's deep patterns in how proteins work, embodied in the billions of different proteins that were fed into this thing. But at the moment, we don't get to understand that.
I would like to see the model be able to be used by certain academic groups. Like, for example, I'd love to use it for improving the activity of programmable gene insertion systems like Stitcher. Like, it would just be like, the tools that we have now are like relatively low throughput. The changes they suggest often don't work. And these kind of powerful tools, I think, would have a huge impact.
And I think that'll happen. Yeah. We just have to, the models, in these frontier model companies like Anthropic or OpenAI, they come out a long time, or they will finish a training and get the model up and running long before they end up actually rolling it out to the general user, because you just want to make sure that it's not going to sort of auto build an atomic bomb or something for you. But I think we'll be able to figure out some kind of program so we can have like really high certitude and trust that the particular people that we'll partner with are going to do like really beautiful things only.
I'm trustworthy, I promise. Let's talk about regulation. I know from the field that there's a lot of, from the community, that there's a lot of dissatisfaction with the fact that aging is not designated as a disease by the FDA, and that we have to go after some clinical endpoint that is a disease and modulate that in order to sort of get our drugs approved. How do you think about how this is going to evolve? Like, is there going to be kind of a GLP-1 from diabetes to obesity to sort of general health and metabolic well-being progression of these aging drugs? Or do you think they're going to sort of remain relegated to these particular sort of clinical indications?
I think fortunately, unfortunately, aging causes lots of disease. So it's not super hard to find proxies for aging in the form of disease, which health authorities know how to regulate. I think it's in some ways like a fantastic structure that we've built for our society to regulate the raw exposure of the pure profit motive versus the average person. It's some of that regulation is good because there are people who will sell you anything. They'll for a little hope if they can make a buck off of it. So I think the discipline of actually making a tangible difference in someone's life, which is essentially in the form of aging, is whether they have a disease or not. I mean, and we can get philosophical all night long about like what exactly is a disease per se, but in the limit, you could reasonably argue that anything that keeps you from doing something that you could have done, say, in your 20s, is technically a disease. Whether it's your muscles don't regenerate well enough, or you have osteoarthritis, or whatever it is. So there's fortunately plenty of diseases to go around. It's also possible, and there already have been INDs issued by the FDA for baskets of diseases that stem from a common aging source. So that's effectively already clinical trials for aging. I think most of the problem right now is just there isn't anything great. I think once there are really powerful, large effect sizes, therapeutics that can work on aging more generally, the regulators will figure out how to get out of the way. Because, by the people, for the people, right?
Yeah, I hope so. So you're in your 60s now. It's coming up on the sort of latter end of the sort of projected human lifespan. Would you be content if your company was successful and you developed some drugs that sort of added 10 years, but sort of you personally didn't achieve escape velocity?
I think for me, it's just an inductive process of like, here I am. I'm very much enjoying 2025. Do I want to go on and enjoy 2026? Yeah. I mean, if you ask me from this sort of manifold here, definitely want to keep going. So, if somebody says to me tomorrow, Joe, you have pancreatic cancer, like, oh, damn, that's really disappointing. We don't know how to cure that yet. And I'm probably screwed, and I don't get to see whatever a bunch of crazy sci-fi stuff that's going to go down. It's probably just going to go faster and faster and faster from now. Okay, so let's say we succeed at this 10 years, and instead of 80 for me on average, it's 90. Say, I don't know. You have to ask me when I'm 89 how I'm feeling about 90, or when I'm 90 how I'm feeling about 91. I have a feeling I'll be pretty into it. If past is prologue, I've enjoyed pretty much most, I guess, of the past years. A fair bit. Good modulo a divorce here and there. It's been a pretty good ride.
Yeah. I think the great quote is that you never die in the past tense. Like you always die in the present. And I think most people just have this like rosy image of, oh, I'm going to be 80 and I will have lived a long life, and I will just sort of kind of rest my head and say goodbye. But in that moment, you're going to not want to die, guaranteed.
Probably, yeah.
Unless you're in significant pain and suffering.
Yeah, if you do want to die, then you could create this sort of synthetic life metric where your will to live is part of being alive. And if your will to live has ebbed to some low, let's say it's 0 to 1, and it's ebbed to like closer to 0, in some ways the dying process already started earlier. You could, if you're like so riddled with pain that you're like, peace out. You started dying earlier, I guess, technically.
Mm-hmm. Second last question for you, Joe. A lot of big generational tech companies leave this legacy of not just like the products and services that they created, but also the mafia of individuals that sort of leave that company and start their own companies and have their own significant impact and generate value for the economy and for humanity. What would you hope the team at Retro will get up to after you've achieved these goals?
Well, one thing I didn't really realize was such a great hack for building a company, again, because this is my third venture funded company, was to build it based on something that is incredibly meaningful to work on, such that if it succeeds, then like the world is just obviously better. I call it a hack just because that's sort of a Y Combinator way of describing things that have like a sort of a clever new twist on how to get things done. Because then people really want to join in because it's obviously meaningful. And that gives you an edge over the other arguably less meaningful things that a lot of people spend, farming clicks basically in Silicon Valley. If that means that some of this ethos just sort of oozes out just as a matter of course for being involved with the company, and it's implicit if you just that we're working on this stuff because it's really important and we like doing it together. I would hope that in the sort of retro diaspora, that people go off and have a bit of that in them. They're thinking, what's the next thing I should build? What if it were tremendously successful would make the world incredibly better? And maybe somebody, if people just keep replaying that algorithm, who knows what will be happening in 20 years. But on the one hand, part of what I consider my challenge and my job is to create more and more exciting, high leverage things for people to do within the context of the company. But companies change sizes, and different people fit different environments. So if they go off and do something, then maybe 20 years from now, like AI viruses will be a plague on humanity or something. Things that eat into LLMs and whatever. It'll make the world a lot better to figure out how to prevent them. Or maybe somebody will actually figure out how to convince humans not to go around killing themselves, killing each other. Back to the ridiculosity of war that we touched on briefly at the beginning of this conversation. Things like that. I think it's a very general heuristic that you can run and just try it out. Okay, imagine this is super successful. What's the world like?
And part of the argument for lifespan extension is that just the very fact that as people project that they're going to be around for the next century and the century after that the sort of time horizons upon which their decisions are made around climate and warfare and etc. Was also extended, that people presumably, hopefully, make better decisions, more rational decisions, more long-termly minded decisions. So
yeah, if people play the long game, I think much more impressive stuff gets built when people's discount rate drops and they're not just living for tomorrow.
Yeah, precisely. If I was a little bit older, I might not be working in the space because I'd be too worried about not enjoying my life and sort of making as much money as possible. Last question for you, Joe. What is one book that you'd recommend to our audience that would presumably inspire them or sort of teach them about the space?
About this space?
Or just anything in general?
So many great books. It's a torturous question. I think one that I often come back to because it reminds me why. I mean, there are a bunch of reminders for me. Having kids was incredible for this. That like humanity is just is like intrinsically wonderful. But weirdly for me, reading David Deutsch kind of gave me a different vision for the long game, for like what humans are and what they can become in the long future. That made it for me, feel a little bit more like really team human, which is a really good feeling. It's a positive emotion for me, and being a little less me and a little more we. Like specifically, the beginning of Infinity as a book,
Yep.
Has a very clear description of what it means to be an explainer. And that's this sort of like rare state of matter, possibly unique in the entire universe, which is matter that can explain the properties of other matter. And it's just really cool, the idea that we're just getting better and better at that, at understanding the universe, and that we'll probably keep getting exponentially better at that. And I don't know, will we ever figure everything out? There's the whole like Big Bang, where did the universe come from in the first place? Definitely very potent mysteries that right now we're just kind of like, maybe, there's a few very not coherent theories, but as we get better and better at and using larger and larger quantities of computation, and probably spreading throughout the universe, maybe we'll figure it out.
Incredible. This is a frame of mind that's shared among many great founders, this like extremely long-term view of the universe. And another great short story would be The Last Question by Isaac Asimov, which like specifically gets to exactly what happens at the very, very, very end of everything. But yeah, on the note of being team human, Joe, thanks for coming on the podcast.
Thanks for having me.
Appreciate your time.
It's a fun conversation. Thanks. Yep.
Awesome.